here at the Institute of Theoretical Physics and SAIFR, ICTP-SAIFR. Ricardo got his PhD at the Institute of Interdisciplinary Physics and Complex Systems in Spain. Then he moved to the University of Princeton as a postdoc. He was also a visiting scholar at the Max Planck Institute for Complex Systems and at the Smithsonian Institute. As I told you, he's currently here. We have this pleasure to have him as a professor here. And his research has focused on studying ecological processes and self-organized structures in ecosystems. And his work has been featured in prestigious journals as Physical Review Letters and Science. And today he's going to give a colloquium, a general colloquium for everybody on ecological systems are not well mixed, the effect of animal movement on population dynamics. Thank you very much, Ricardo.
Thank you, George. Well, it's my pleasure to not only be here today, but also to work here. I changed a little bit the title because I realized that saying ecological systems are not well mixed might be a little bit aggressive, and it's always not good to generalize, so I said they are rarely well mixed. But the spirit is more or less the same. So the talk, I usually don't give very technical talks, but I know the kind of audience that I have today, and I know that most of you guys really like math and equations, so I try to find a midpoint in which I'm going to present the project that I think is more technical of all of the things that we are developing here. But I didn't prepare so much detail, but I have a lot of slides with the math and how do we do the things. So if you are interested in one of those, we can either in the question time go through details, or you can find me whenever and we can discuss the more technical parts if you wish. I don't know that most of you are not very familiar with the questions that we ask and we answer sometimes. So I also wanted to give you a broad overview of what we think and show you systems in which all the things that we know as physicists might be useful. So this is the kind of twofold goal that I have today. So most of the work I'm going to present is joint work with these 4 people here in the screen for a long time. So we started working on all these things when I was in Princeton like 6 years ago. So it's, well, Bill, Fagan, and Chris, they are in the University of Maryland in the US. Mike is in Canada now, in University of British Columbia, and then Justin Calabrese, he's a professor in Kassel in Germany, who is a Helmholtz Institute. They used to be all of them in the Smithsonian. That's why we started working together on this. And some of the ideas that I'm going to, yeah, this is the thing that takes a signal. Most of the things that we started developing here in IFT in Brazil are done by some of the people that I work with. With Benjamin, he's a master's student at IFT. Rafael, he's a PhD student in UNESP that also works with me. And then Vivian and Paulo, who are postdocs from SAIFR. So I'm not going to go into a lot of detail on some of the work that Vivian and Paulo are doing, but this is more or less the people that are developing the projects that I'm going to discuss today. There's more people I have pleasure to work with here, but I couldn't explain it all and not lose everyone. But before we go to, I start being more specific on the projects that I want to discuss, I want to explain you a little bit the approach that we take in the research that we do when we study ecological systems using physics and the kind of systems and the kind of questions that we address. So basically, we focus on trying to understand how ecological patterns that we observe in nature emerge from interactions that are happening at a level that, let's say, it's below the level at which we are observing this behavior. This is a little confusing because it has level, behaviors, interactions, but it's going to be very clear in 3 seconds. So basically what we do is we try to understand how given individuals, for instance, the members of a population, a collection of cells, a collection of animals, how they interact with each other and how they interact with the environment, with the place where they live, with the sea, with the river, with the forest, with the temperature, wherever they live. And we try to develop models in which we understand how these interactions are going to affect the way these entities move and reproduce, which are 2 of the main things that living things do. This is the first thing we have to learn. and how these interactions result in this pattern that we observe, this larger scale structure that we observe. And then using those models and using data that we get very often from collaborators, we are not lucky enough to go to the field ourselves, we try to understand what are the consequences at ecological and evolutionary scales of these structures. So this is kind of the approach that we follow. It's very abstract. So what I'm going to show you is a little bit of systems in which we work. So now we have some videos before we go into lines and equations. So one system that we've been studying for quite some time now is the creation, the formation of spatial patterns in bacterial colonies, in microbial systems in general, in microbial colonies more specifically. So what you saw in the screen was an experiment that one of our collaborators in the US did, in which basically we took two strains, two types of the same bacteria, that they are identical, but we manipulate them genetically. So one of them is red and the other one is blue. We throw these bacteria in a microfluidic chamber, which is an experimental device, a surface, and we allow them to grow, and then we study how, depending on the initial density of bacteria and depending on the intensity of the resources that we provide them, how they colonize this surface. This is experimental data, and then we develop models based on lattice systems like the EASY model, the contact process, to try to understand both the characteristic cluster size of this pattern, the roughness of the surface, so whether we can find some universality class in the roughness of these frontiers between clusters and things like that that are more physics questions. We also study microbial systems in another organism. So what you see, and let me stop the movie before I spoil it. So what you're going to see now in the screen is all these small dots. They are cells, individual cells. They have food, so they are very happy and they are moving around and they are dividing. But at some point they will run out of food, and what they are going to do is they are going to start communicating with each other through chemicals, and they are going to form these aggregates that you can see now as darker points. So we study how this communication process between cells is driving this aggregation, what is determining the size of the aggregate, the formation of these territories, and so on and so forth. So we do, again, experiments and models to understand this system. The models are based on random walk theory, PDEs, and things like that. So random walks is something that we do a lot. The other system we work with is much larger. So this is the wild miss migration that probably many of you saw in National Geographic. So these animals, they move across the African savanna every year in a very specific way, in a very closed loop. And basically, they are somehow able to feel that the environment is going to change and that they have to move from north to south, from south to west, from west to north, and they repeat that every time. When they do that, they interact with other species, they manipulate. Their migration is actually manipulating the ecosystem in very interesting ways. The easiest one is because you can imagine that when you have hundreds of thousands of animals moving around, they throw stuff in the floor, they eat grass, they do their stuff, they secrete poop, for instance, and that enhances the microbial activity of the ecosystem. So we try to understand these processes. And then, before I move to more specific results I want to discuss, another system that we study a lot, and actually we have been working on this for a very long time, is the formation of these structures of vegetation that we observe in aerial images in many regions in the world. So what you see here is, this is actually Google Maps screenshot. So this is a region in Australia, and you see that the length of this white bar here is 10 meters. So basically what is happening here is because of the lack of water, plants are interacting with each other to try to obtain as much water as possible and survive. And in doing so, they are creating this pattern that looks like a honeycomb of holes in the ground. Depending on the availability of water and depending on surface properties, the shape of these patterns may change in very different ways. And basically what we do here is we develop reaction diffusion models based on Turing theory to try to understand the interactions between water and vegetation and how these plants are creating these structures. So here's something that is very interesting, is that this is 10 meters, and this is grass. So it is not that, I mean, there's actually some collective interaction between plants to create these larger scale structures. Anyway, this is a bit of an overview. If you want to learn more about any of these, we can talk, of course. And you can also, we recently published this review paper where we cover all these self-organized structures in very different systems, and we have a lot of pictures. And I don't think there is any equation in this paper, so it's a nice reading for, I don't know, the bus or the metro or anything. Great. So what I really want to talk today is a much general and a much theoretical problem, which is population dynamics. So basically, you can imagine that when these entities are interacting with each other, the population is changing. And this has been studied for a very long time, mostly using ODEs. And I realized when I arrived here that many people are annoyed with the use and abuse of ODEs in mathematical modeling. So I don't know, I was talking to students that already left, and they were thinking that using ODEs was a too strong assumption. And it is true, it is a too strong assumption. It's super useful. It's been giving us a lot of insights to understand how these populations behave. But what we really are trying to understand is to refine these models based on ODEs. So basically what I'm trying to do is not to annoy people here with models of population dynamics. And the claim that we do to take this approach, and the first thing that I want to say to try to provoke a little bit of thought, is that this population dynamics is actually an emergent phenomenon as well. So when we see the number of rabbits changing in Australia for the last 36 years, if we think carefully about that, that is an emergent phenomenon. It's still emerging from how rabbits are eating grass and how they are killing each other, and there is some underlying process behind the scale that we are observing that is determining that pattern that we observe when we see number of rabbits through time and we see a line. So I'm not going to talk about rabbits today, sadly. They are very cute, but I'm going to talk about something that is more important than rabbits, which is yeast. And the reason why I think yeast is very important is because it's responsible for bread, wine, beer. So no yeast, no party. So there is this experimental result. It's a classic experiment in ecology from the 30s, in which basically what Gauss did was he took some cells from some species of yeast, he put them in a growth media, he gave them food, and then he tracked through time the amount of cells that he had in his bottle. And what he found was this. So he saw that in the first phase, the population was growing, and then eventually the growth saturated. He repeated this experiment with different yeast, and he observed a qualitatively similar pattern of population growth. Of course, the scale here is not the same. The scale here is not the same because the parameters are not the same. The growth rates are not the same. But you can say, well, it first grows and it then saturates, which I think is a fair statement. Then, of course, these results, I have a variable that changes with time, so I can model it with an ODE. Why not? I mean, it makes perfect sense. So that is what people started doing, and then it's when we have probably the most famous equation in population dynamics, which is the logistic equation. So basically, people thought that this pattern of population growth has two kinds of behaviors, as I was saying. So in a short time, the population is very diluted, and then these guys are just growing exponentially, and that's what is giving me this linear term. If I solve this equation, I have an exponential solution. But then eventually, as the population gets larger and larger and larger, there is some limitation to the growth, and that is represented by the quadratic term that dominates when n is large, and is actually what balances the growth of the population. Then, of course, parameterizing R and K, fitting R and K, you can try to get how different species grow and the temporal scales and so on and so forth. So that is, very simplified, a way in which people have been understanding these population dynamics problems for a long time. My claim today is that actually, what we can do as well is to try to look a little bit closer into these cells and try to understand this macroscopic pattern of growth, really looking at what is happening microscopically. So we can try to describe a microscopic individual level of the processes that are happening there, and then we can use tools that we know well from statistical mechanics to try to get these macroscopic, these equations, these coarse-grained equations that are describing the macroscopic behavior of the system. This is basically what one can do to obtain these ODE models that mathematical biologists started postulating more phenomenologically in the 19th century. So we can write down a model that now, instead of being an ODE for the number of cells that I have, it's going to describe the processes, the stochastic processes that these cells are suffering, which are going to be birth. Of course, if a population grows, something has to reproduce. Death, that I'm going to represent by this kind of notation in which I have one cell that then goes to 0 at a rate d. And then, in order to account for this limitation of the growth, I can write down another reaction that is going to be is going to represent some sort of the collision of two individuals, the encounter of two individuals, and the death of one of them as a result of this collision. And that is going to happen at a rate gamma. Given these rates, I can write a master equation for the probability of having n cells at time t. We all probably know how to do that. And then, if I am able to solve that master equation, I will have the full solution of my problem. I will know what is the probability that I have n cells at every time step. So I have a stochastic description of this process. And of course, I can manipulate this master equation if I am not able to solve it, which many times is the case. And I can obtain, for instance, an equation for the mean value of the number of cells that I have in my population that is going to be the logistic model. So somehow this logistic equation that mathematical biologists postulated, and that many times you will see it's written down like, this is a logistic equation, x dot equal to rx minus. It can be derived in a more formal way, starting from a mechanistic description of what is happening here. Okay? But this, when we start thinking a little bit on this, and then of course I can do things like, okay, now I'm going to obtain the equilibrium of this ODE, and I'm going to set the time derivative to 0, and I'm going to find the value here at saturation that is going to be B minus D divided by gamma. So I can recover all the results. This is the take-home message from this first part. I can recover all these results that may sound more hand-wavy and more phenomenological, starting from a mechanistic description of what is happening there microscopically and using statistical mechanics. Great. How useful it is? Well, if you are a physicist, you like doing it. Biologists often don't like it. But the reason why we believe it's extremely important to follow this approach is because it's only when we do that, when we really think, and when we spend time thinking about the assumptions that are behind these models, right? So probably, I don't know, well, now I spoiled myself, but when I was showing you the logistic equation, this quadratic term, I would bet that many of you said, this makes sense. I need some limitations, so then I need a quadratic term to balance the exponential growth, and that is it. But I don't know how many of you thought on what I am assuming when I say that that is a quadratic term. Well, if you work on collisions, law of mass action, well, maybe we start thinking that I am following some sort of law of mass action. But I can tell you that is not certainly the case of many people applying this equation. So there is no so much thinking about the microscopic processes that are happening there, and then we don't know what are the limitations, basically, of the models that we are using if we don't know how to derive them. So this logistic model, and any model that we see in ecological theory for many, many years, it relies on this law of mass action for ecological interactions. So basically, again, microscopically, all I assume when I wrote this encounter reaction is that these cells in my bottle, they are going to be like ideal gas particles, and they are going to be all over the place, and they will encounter with each other by chance, randomly. Of course, that for the gist, maybe it's true, but we'll see that there are places where it is not true. So basically what I am assuming is that these individuals, these cells are following a trajectory that is described by Brownian motion. So it's the C is going to be the position of the individual, 2 coordinates, x and y. G is the amplitude of this stochastic force that is basically white noise. It's an uncorrelated force. Sorry, I don't know how much of a stochastic processes, because everyone that took my course on stochastic processes is not here. So that's why sometimes I get too basic with stochastic processes. You can interrupt me if I am going too fast, or if I'm going too slow, you tell me, this is very easy, move forward. Great. So the point is when we have Brownian motion, we know that the variance of, of this variable is going to grow in time proportional to t, and then basically, if I don't put boundary conditions, my Brownian particles are going to be all over the place in the long term. So I also need to introduce some reflecting boundary conditions, and when I do that, in the long term, the probability distribution function for the location of these cells, of these individuals, is going to be uniform. And it is then when I recover this law of max action, because I basically have a collection of cells, particles, individuals, that are equally likely to be everywhere in this environment, and therefore the collisions may happen randomly. Okay? This is what I'm going to call a well-mixed system in the next, I don't know, 30, 40 minutes. So what is the problem, and what is driving our research question? The problem is that nowadays, we have access to data that allow us to understand how these animals move in space. And basically, the way we collect this data, this is data for wolves. We go there to the field, we put them a GPS, a collar that has a GPS, basically what Android, Apple does with us. And that is recording the position of this animal through time. So we have the trajectories. When we observe these trajectories, this is how they look like. This is 6 wolves that you will see moving, I think it's in Canada. And you will see that this is everything but well mixed. It's everything, I mean, it's very different from what we were seeing before. I have the white wolf is much more likely to interact with the blue or the red than with the purple or the pink or whatever color this is. So these populations, because of the way they move, they create spatial structures. So then the follow-up question that we have is, okay, so then how is this structure going to determine or to modulate what we were inferring when we were assuming that they were all over the place, okay? Great. So to do that, I'm going to answer 3 questions, if I have time, if not 2, and if not 1, that are related to understanding how these realistic movement behaviors are going to reformulate existing results on ecological theory. Of course, I'm going to start very simple. I'm going to start with, yes?
Concerning your previous slide, those colors are related to groups of wolves or individual wolves?
Yes. So here what we are doing is we are tracking one individual from each group. Okay. So these wolves, they live in packs, so they live in groups, and we are tracking one individual from 6 groups. Well, we know these people, yes. Yeah, so there is levels of organization and groups, and things get much more fun than what I'm going to discuss. But also that gives us, hopefully, work for the next 20, 30 years. Great. So this is the question I want to address generally, and I'm going to break it in parts. The first part is going to be with n equal to 2. Sorry if I disappoint anyone. So first of all, I'm going to try to derive results for the probability of 2 individuals that are moving according to this realistic behavior to encounter with each other. Great, n equal to 2. And then I'm going also to introduce the model for not well mixed. And then I am going, once we have this new theory for ecological encounters, I'm going to show you one application to data. So you see how this theory translates to the real world. And I am also going to discuss the application to a very important problem that we have nowadays, which is how this behavior affects the way animals interact with humans, in this case with vehicles. And this is Benjamin's master's thesis. And then I am going to discuss, I want to wrap up. I started with the logistic equation, so I want to finish by telling you how this logistic equation gets when we introduce movement, which probably will give you an idea of how the logistic equation was and is used in so many applications. Great. So let's start with the beginning, with this encounter theory. The reason why I want to start with the encounter between two individuals is now, I mean, of course, we always want to start with small population sizes, with small systems, because that's the way we can get more insights. But it is also because these interactions between two individuals are, let's say, the building block of a lot of ecological processes, not only population dynamics in the sense of competition, as you were seeing in these experiments, but also in reproduction, mating. So there is no mating if two individuals don't find each other. Also predation, predator can only eat a prey if they find it. And as we sadly got aware recently in disease transmission, no? So all the measures of staying at home and so on, we're trying to reduce these encounter rates, basically. Great. So how are we going to refine this well-mixed hypothesis? So I already said that we are going to work with n equal to 2. And then I need to develop a model for how these animals move that creates this spatial structure. And this is going to be nothing else but a harmonic oscillator. So, and this is not, I don't know if actually we're physicists the ones introducing this model into movement, but basically what we are going to assume is that these animals, they moved trapped in a harmonic potential. And that harmonic potential is basically what is going to confine the trajectories of the animals and is what is going to avoid the unbounded growth of the variance in the trajectories. So now I'm going to modify my movement equations. You see that this term here is the one that we had for Brownian motion. And I'm going to introduce a deterministic drift, which is basically a linear force that is going to drag the animal towards some position, some location lambda, that is going to be the nest, the center of this territory. So basically, you can think about this movement model as my wolf being translated to a particle that is trapped in a harmonic potential with some thermal energy that is driving these fluctuations in the location around this center. Okay, this is what this movement model translated to physics tells me. Great. So now, of course, this is the white noise is a Gaussian process. And I am only adding a linear force, and that means that then C, it's also a Gaussian process. So I can describe it completely if I give its mean value and its covariance matrix. Okay, this is theory from stochastic processes. I can calculate it because this is a very simple stochastic process. This is the Ornstein-Uhlenbeck process. So I can analytically obtain the covariance matrix and the mean value. And here I'm assuming that x and y are not correlated. So basically the movement in the x-axis is not correlated with the movement in the y-axis. That is why these non-diagonal elements are 0. We can extend the calculation assuming cross correlations, assuming correlations, and things don't change too much. So I prefer to keep it simple. So yes, so C is a two-dimensional vector with x and y. So the notation sometimes is too explicit because, well, I usually communicate with people that have different levels of math, so this kind of notation helps. But of course, this is g tau over 2 multiplied by the identity matrix. So what is interesting is that now, as I was saying, because of this linear force, even if I integrate this equation, this stochastic differential equation I integrated with the computer, I simulated, if I do it for, I don't know, forever, for a long, long time, the trajectory of the animal is now not going to cover this long territory. The variance is not going to grow indefinitely in time. It's going to be bounded, and therefore my trajectories are going to be trapped within these regions of radius rho that I am going to define as the home range, as the radius of the territory of these animals, okay? Basically, this rho, you see that the trajectory leaves the rho from time to time. This is because now these they are not boundary conditions, and this is very important. Before I was imposing reflective boundary conditions, but now all I'm doing is I am defining this rho based on the percentile of the distribution. So basically I'm saying that this rho is the region of the space that will contain 95% of the probability distribution function of my individual. So of course these trajectories can leave the boundary, the territory. That happens also in data, but this is the standard way of defining these territories. Great. So just summarizing a little bit the comparison between these two movement models, the key idea here, and this is what I want to, all of you to remember, is that when I compare the two movement models, the Austin-Uhlenbeck process, the one that contains this linear force, is introducing an additional time spatial scale that is actually going to define these Hom ranges, and it is the one that is going to define the range of the interactions. So now the interactions between this gray guy and wherever other individual I consider here, they are not going to happen anywhere in this territory, but they will be restricted to the overlaps between these territories. And that is the question that I'm going to address with the calculation that I'm going to show you now. The question is how the existence of this new scale is going to influence the probability of encounters. Okay? Great. So this is basically the idea. The idea is I'm going to compare a case in which I have a blue guy and a gray guy that can encounter anywhere in the island. Let's assume that this is my hometown in Spain, the Canary Islands. And on the left, we have that these two individuals, they don't share the whole territory because of the way they move. So therefore, the encounter probabilities are going to be different, and I'm going to calculate that encounter probability. Okay? So again, I am not going to do the whole calculation. I'm going to go through the steps, and then if you want, we can even use the Blackboard and do all the calculations, so you can do it as homework, and then we discuss it as you wish. So the first thing I have to do is I have to define this probability of encounter. So I'm going to define the probability that I observe an encounter between a time t and a time t plus dt as some encounter rate that I'm going to call this capital epsilon times the time interval at which I am observing this encounter. And all I'm going to do is I'm going to calculate this encounter rate. So this encounter rate, and this is standard in the literature, I'm going to assume that it has two components. One of the components is going to be spatially dependent and is going to tell me, given a distance between two individuals, how likely it is that an encounter happens in a given neighborhood around one of them. So imagine that I have here a rabbit, and I have here a fox. So I'm going to define the encounter rate between the rabbit and the fox with some spatial component that is going to give me what is the probability distribution function that I observe this encounter in a neighborhood of the predator, sorry, times some gamma parameter that is going to be the attack efficiency. So imagine that the gamma is going to be how good the fox is at killing the rabbit whenever he goes for it. And this phi is going to be this probability distribution function that we can fit. I'm going to use a uniform distribution, but we can extend the calculation to any phi, and things, again, don't change qualitatively. So, of course, if my encounters are probabilistic, the encounter rate is going to be also a stochastic process. So imagine that I have my two trajectories that I'm going to simulate in time, and I track this epsilon, and sorry, epsilon there should be a function of, and I track this epsilon as a function of time. It's going to be 0 when the two individuals are very far, and then it's going to jump to gamma when they are close, and it's going to be a sort of random train of spikes. So this is going to be stochastic as well. So all I'm going to do is to take the mean value of that stochastic value, of that stochastic process, and I'm going to calculate the mean encounter rate, which is basically the probability per unit time that my two individuals will encounter with each other as I average over several realizations of the movement trajectories. Okay, so I'm going to run these two trajectories 10,000 times, and I'm going to record these probabilities of encounters. I'm going to take the average at every time step. That would be computationally. I'm going to do it analytically. Yes?
This is an instantaneous rate counting. You calculate it with respect to the, let's say, initial position of the other, or the prey, let's say, or you calculate it to the actual position in time.
The actual position in time. Yes. So this is a function that tells me, as a function of time, how likely they are to encounter. And because of the way I am defining this phi, it's going to be 0 when they are very far, and it's going to be a constant when they are very close. So basically, all I'm doing is defining a function that tells me, as a function of how far the two guys are from each other, how likely they are to encounter each other. So I always have a lot of problems to introduce this. Yes?
Maybe a meta question, but I mean, I understand you can compute this. So you,
Yes.
you wrote the problem, but you can compute the answer to it. But
Yes.
I don't understand what problem you're trying to solve. I mean, you started with some equation, and you say, well, this is the equation I'm going to start with. But what does that have to do with the animals? I'm missing it. If I tell you foxes or I tell you snails, you can use the same equation.
So maybe it's, I don't know when you arrive to the talk. Okay, no, so maybe it's because of that. No, but it's fine. We have time. So the idea is that, for instance, snails or frogs or your favorite animal may show patterns of movement that make these populations to be spatially structured. So,
No, I'm just asking the next transparency.
which one?
The one with the Uhlenbeck formula.
Yes.
You wrote down that formula, but I
Yes.
don't understand where that formula came from.
Great, so this is a model. So it's an equation that I propose for modeling the movement of the animals. And I can give, depending on the species, G and tau are going to be different.
So I'm happy to start with that. I just don't see why you started.
So I start with it because it reproduces the movement behavior of, I have a data set with movement behavior of 600 species. And this equation reproduces 554. Okay, great. Depending on tau and gamma. Great. And to refine it, you need to introduce autocorrelation in the velocity, so you can also improve it. But yeah, that is the idea. The idea is that you can fit this model to explain movement behavior of many species. Great. Anyway, all I'm doing is, if I have my prey, and this is much better than this. So if I have the trajectory of the two individuals, all I'm doing is this phi q is going to give me some distribution of risk around the wolf. So how dangerous it gets for the rabbit when he is around the fox as a function of the distance. And then the gamma is telling me how good the fox is killing the prey when she's in the red region. So basically, for instance, in a configuration like this, epsilon would be 0. But in a configuration like this, epsilon is going to be gamma, because they are close enough, and now the probability is actually gamma times the time interval, because they are close enough. Okay. So again, you can start to figure out that a key quantity to perform this computation is to understand how this pairwise distance, dr, between the two individuals, how it behaves statistically. So what is the distribution of that variable? So you can calculate it, and you do all the calculations. Again, I am happy to open it later. But what is important when you obtain these encounter probabilities with the two movement behaviors that we had is that in the case of the Austen-Uhlenbeck movement, the encounter rate depends on many spatial scales. In this case, it depends on the territory size of individual 1, the territory size of individual 2, and the distance between the centers of the territories, which is this R lambda. So it basically depends on 3 spatial scales, whereas here, in the law of mass action, in the results that we know for a long time, it only depends on 1 spatial scale. It only depends on the size of the box where these animals are contained. So yes? Yes, so basically the way I did this calculation is I did it for a general Q, which is the size of this red thing, and then I took the limit Q to 0 because usually the range, so this Q, you can understand it as a perceptual range of the animal, so how far he's able to see. So this Q is usually much smaller than any other spatial scale in the system. It's much smaller, much shorter than rho, that the size of the territories, of course. I mean, in some species not, but in general. And then the distance between these territories. So basically, I then took the limit Q going to 0. If you don't take this limit, the difference is not very important because Q appears in both expressions. So instead of 1 and 3, you have 4 and 2. But still, the difference is 2 additional spatial scales. So once we get to this result and we start interpreting it in terms of the geometry of the problem, I think it can be quite, I don't know, at least my initial intuition was, well, I mean, this makes sense. I have these guys that they move, I mean, they have territories, and then the encounter depends somehow in the overlap. On top of that, the distance between the home range centers enters exponentially here in the equation, so we can think that the encounter rate is going to decay dramatically as these guys put their territories apart. So in general, I can think, okay, take home message, when I assume a spatially structured population and I introduce realistic movement behavior, the encounter rates are lower than in the well-mixed, and then I basically, wherever I get with the well-mixed hypothesis, it's overestimated with some factor, and I move forward. I take that for granted. But that is not true. When you compare this result with the results for Brownian motion, which is basically what I'm showing you in this plot, is the ratio between the OU and the Brownian motion encounter rate, you see that depending on the parameterization of the territories, depending on the composition of the spatial configuration of the population, when you assume Brownian motion or well-mixed populations, you can either underestimate or overestimate the encounter rates. So this goes in two directions, and therefore it is important to stop a little bit and try to think how these animals move before we make estimations on how likely they are to encounter, which is related to disease spreading probabilities, probabilities of having an outbreak when we have some parasites in a horse population in wherever. So it's also translated to practice, to people that are trying to understand populations and to manage problems with those populations. And there is no way that we make a one-direction guidance in the sense of, oh, be careful, you are overestimated by a factor exponential of the, no. So it's context dependent, which makes things trickier. Of course, the intuition behind this, I think once we stop, yes?
I didn't understand really your figures. Can you explain a little bit better?
Yes. So basically what I'm representing here is the ratio between the epsilon that I calculated. Okay. I'm representing the ratio between this epsilon and this epsilon, one divided by the other. And I am doing that as a function of the R lambda, the distance between the territories, and the home range of the predator. So basically, I think, okay, so basically what I'm doing is I am considering that my predator has this territory, for instance. And then, actually, this is what I'm doing. I am considering that my predator has this gray territory, and my prey has the blue one, the smaller one inside. This is the typical behavior between the territories of prey and predators. Usually predators have much larger territories. And then all I'm doing is shifting where the prey territory is located inside the predator territory. And for each of these configurations, I'm computing the probabilities, the ratio between the encounter probabilities. And what we observe is that when the distance between the prey territory and the predator territory is small, then the OU encounter rate is much larger than the well-mixed encounter rate. Whereas as we shift apart the territory of the prey, the encounter rate of the OU movement decays, whereas the one of the well-mixed system does not. And therefore, there is this transition between overestimation and underestimation. The idea is even simpler. Basically, when I study a OU encounter process in one dimension, this is what I have. I have two normal distributions. For the predator, I have the gray one because it's broader, has more variance. And for the prey, I have the blue one. We can think that encounter rate, it is not true, but we can think that it is related to the overlap between these two. Well, it is true that it's related. It is not true that it's a one-to-one connection. So basically, as I move the blue distribution, I am changing the overlap, and I am diminishing it. Whereas in the case of the Brownian motion, it is always the same, because the overlap is the overlap between two uniform distributions within my island, and that is constant. So this is the reason why the encounter rates, when we don't model movement realistically, can be either larger or smaller than the real ones. Okay? Great. So this is the theoretical result that we derived for encounter rates. And this is, well, the reference will appear at the end if you're interested. But Nathan actually was asking something that is very interesting. It's like, why did you start with this equation? And we start with that equation first because it's one of the models that is most used and is more successful in explaining animal movement. But if we want to continue this dialogue between justifying the choices that we do, not only for the joy of math, but also for bringing things to reality, remember that we are trying to understand natural systems, we need to show that the theory that we are developing can be used by ecologists to answer the questions that they are interested in asking. If not, well, mathematicians will like it, physicists, I don't know, and biologists will not care about it. So the way I want to show you how we connected this new theory with an actual tool that can allow us to understand populations is with the second ingredient on how real movement changes the encounters of the animals. So when I show you the data of the wolves, and we saw these 6 territories from each of the packs, I told you, well, the blue guy will very likely interact more with its neighbor than with the other one that is very far apart. So basically, this spatial structure is going to change 2 things. It's going to change the temporal statistics of the encounters, and we already saw it, but it is going to change the spatial statistics as well. So there will be regions of the space in which my encounters are going to be more likely to happen than others, related again to the overlap. So we can obtain analytical expressions for the probability distribution function of these encounter locations. This is something we did in this work here. And then we can develop statistical tools that will allow, and actually do, they allow people to introduce their data, their GPS tracking data, and will return them a spatial distribution of where their animals will encounter with each other. So basically, we can develop a statistical tool that is going to have as an input these 2 trajectories, the gray and the blue, and as an output is going to give you this red probability distribution function that you see here. Okay? And then we can compare it with actual encounters if we have recording of encounters and so on and so forth. So why is that interesting? That is interesting because we can use those statistical tools to learn something about the systems that we're studying, to learn some ecology. And ecology in the beginning seems boring because it's messy and it's dirty, but as you get familiar with it, it becomes more interesting, at least to me. For instance, if you work with ecology, you have the chance to see these nice monkeys in Panama. So that is something that you don't see if you don't work with, well, now you see in the picture, but you can work with them. And actually, we had data on these monkeys as well. The data set that we used for this was collected by 2 of our collaborators, Grace and Meg Vegan. They used to be in the Smithsonian Tropical Research Institute in Panama and in UC Davis. Now they are in Germany. And basically, this is how the data looks like. So we have data. Again, these monkeys are very similar to the wolves. In behavior, they are very social with the guys from their group, but they are very aggressive with guys from other groups. So what you see here is, again, 1 monkey from from different groups that we track through time, and you see their trajectories in different colors. I don't think I have a movie for this, but yes. So here what you see is that the monkeys are basically following their, well, the trajectory that we had, but again, they establish territories. So this is a perfect data set to see how good our tool works. So basically, if we provide this statistical method with the trajectories of these 6 animals, it should give us some distribution that somehow should be very high in these borders, and it should be not so high inside the territories. So this is a different type of plot, but this is basically the same data set. As you see, this should be the territory of one of the monkeys, and the red contour that you see is, again, the 95% percentile. So this region inside the red contour is the region of space in which 95% of the encounters will take place. Not surprisingly, it is in the border between territories, but you can also use this tool to switch your question. I mean, if you don't have very high resolution data, you can use this to understand where are the borders between territories as well. So why is this important for ecologists? I think we only have one ecologist in the room. But this is important because we can try to understand things related to the behavior of these monkeys. So I told you, these monkeys are aggressive. They don't like finding a guy from another group. I don't know, they support Palmeiras and they don't like finding someone from Corinthians. How can we measure this in data? We can try to see whether we find different patterns of movement inside and outside this region. So if I don't like finding another guy, whenever I get into their neighborhood, I will behave differently than when I am in my territory, because I will feel danger. So what we did was to split the data set for each of these monkeys, and we analyzed the movement behavior, the mean velocity, inside their territory, and outside the red region and inside the red region. And I don't know why. And this is what we found. What we found is that inside the red region, the average mean velocity of the animals is significantly higher than inside, and also each of the individuals itself is moving significantly faster than all the others. Well, we have here 2 exceptions. Probably these are, I don't know, they like action. Anyway, so this is an example of how we can apply the theory that we developed, so this calculation with pairwise distance distribution and integrals, to actual tools that can be used by people that work in the field and are interested in understanding, in this case, how a monkey population works. We also apply it to lizards and to simulation data, and this is the reference for this. Great, so I don't know how I'm doing in time because I speak a lot. I think actually 5 minutes? Okay, so I'm going to go extremely fast through, okay, so then I'm going to be very fast. This is very quickly. So basically, I wanted to, also because when I came here, I was very lucky to get a lot of resources, and I wanted to show how we use these resources. I think it's very important. So this is basically Benjamin's master's thesis. I'm going to try to explain it as good as he would do. But basically, what we are trying to understand is how this real movement behavior is going to change the probabilities and the number of deaths that roads can cause in a population of animals. And this is very interesting mathematically because this is a mean first passage problem. I don't know how many of you have read about mean first passage problems, but basically means that instead of calculating the probability per unit time that these guys will find each other, we are interested in finding the time at which they encounter for the first time. And where do they encounter for the first time? And that is mathematically way more challenging. That's why I did the other part and Benjamin did this, because it's way harder. But we can still do it. So basically what we are going to have is an animal. So the other plots were done by ecologists, and this is done by me. So we have an animal, and we have here the center of its territory. It's going to be at a distance a from a given road, and the animal is going to start from some x0 initial distance to the center. Anytime the animal crosses this road, it's going to have a probability pk of being killed by a car. Okay? So all I'm going to do is I'm going to study these trajectories, and I'm going to calculate the first time at which this animal is going to die. So these first passage problems using OU models are extremely hard. Actually, there is no analytical expression for the mean first encounter time between two OU processes in 2 dimensions. That is a non-solved problem. There is no analytical solution for that. But for the road problem, we can use the symmetries to map it to a 1-dimensional problem, and then we can solve it in 1 dimension. Basically, you can think that if the animal moves isotropically, then the y coordinate is not important, and everything happens in x, and then we can do it in 1 dimension. And then we get a nice and closed expression for the mean first passage time, which is this one here. I can discuss the limits and the asymptotics if you want at the end. But basically, well, you see that it agrees very well with the simulations. And then we can basically, as I said, try to understand here the key parameters in which we recover the Brownian motion limit. We can do that, and we can compare it, and we can do the same analysis that we were doing before. So again, all what I want you to think is that we can incorporate this realistic movement, and we can refine a lot of theory to try to understand a lot of applied problems that at the end they also become open. I mean, you also find in a way open problems in statistical physics. So it seems that Benjamin now is trying to solve this in 2D. He likes challenges, and hopefully we get it done soon. So yeah, I think I don't have time, but I just wanted to finish with the logistic things, because I wanted to close the story. And basically, and this is Rafael's PhD, basically when we discard any spatial effect, everything is an ODE and everything is pretty. But when we have a spatial structure, basically what we have is a dynamical process in which we actually have to also track where individuals are, where do they go after reproduction. So we have a dynamics of a population that is happening in space. So the way we can write the master equation now, it is not for the probability of having n individuals at a time t. It's a master equation for the distribution of individuals in space. So it's a spatial distribution of animals. And of course, the statistical physics tools that we are going to use, they are much harder. They are related to if we define the probability of the spatial distribution of individuals as a sum of deltas. So whenever I have an animal, I have a delta. Then, of course, I have to treat this distribution using the spatial moments of this distribution, and then things basically get extremely messy. And basically, the take-home message, and with this I finish. I have a lot of slides. If you want me to then, wherever, go slowly, we can. But basically, the take-home message is that if you remember before, we had that the population size in the equilibrium was B minus D divided by gamma. Now, when I account for space, I have a correction term that depends on the per-correlation function of the population. So this correlation function is what is containing all the spatial information of this population. When the system is homogeneous, the correlation function, which tells me the probability that I have 2 particles at a given distance, that is going to be 1 because of the normalization for every possible distance. And therefore, this integral is going to be equal to 1 because this is normalized, and I recover the result that I had before. But as I have some emergent spatial structure, this integral is going to be different to 1, and it's going to correct in different directions the population size. What we are trying to do now, and sorry I was very fast here, is to understand how the movement is determining this correlation function here. And I think this is a problem that can be very interesting for people that are very good at field theories, because at the end, all we have here is the annihilation and the creation of particles at different spatial locations, and all we are trying to do is to do some statistical field theory to the master equation. But that is something that we are trying to do now. So I'm watching some classes that they are in the IFT YouTube channel by Pedro on the statistical field theory. So hopefully I understand that and we can apply it.
Excuse me, that function C will be the one that essentially, in the case of the predator and prey, will pass information between the let's say, elements of a given community that the predator is, in a way,
No, but it is for a single population. So this per-correlation function is related to the structure factor of a system. So basically, if this is our population, and because it is discrete, I have a spatial distribution of points. So this per correlation function is going to tell me is going basically to tell me for every pair of individuals what is the probability that they are at the distance vector t. So basically, if my population is homogeneous, because of the normalization that I am considering, this should be equal to 1 for every distance. When the and this is the green line. When the population has short range aggregation, for instance, because of the way individuals move, it has some clustering, then the correlation function at short values of chi is going to be larger than 1, because it's more likely that I find pairs close to each other. And when they have over-dispersal, for instance, in a prey-predator system, you see that when you calculate the correlation between the location of the prey and the predator, because the prey wants to run away. So in that case, well, I didn't find any configuration there, but basically the C would be smaller than 1. So this would be dispersed, this would be aggregate, and 1 would be, let's say, well mixed. So it's a way of characterizing the spatial configuration of the system. And, well, sorry, with this, these are the two foundations that supported all my work. Well, these are two of the foundations that supported my work here in Brazil. The papers that I discussed. And if you want to read about the ones in the beginning, this is the list. And if you have questions, I'm happy to take them. And thank you.
We'll have time for some questions. So it seems, Nathan?
So, sorry, going back to this Uhlenbeck equation, I just remember Uhlenbeck, I don't remember the first one. What kinds of animal species or animal traits do you need for that equation to be satisfied? I mean, it's
And
not
for the Olsen-Uhlenbeck?
Yeah, do we have to have herds or something like that? Or I didn't understand exactly.
No, it's quite general, because all you have to do is, so very general, you can classify the movement behavior, you can classify the behavior of animals in terms of the movement in the long term as nomadic species, for instance, the species that move very large territories through their lifetime. You also have migratory species. So for instance, I will go to your answer, but I need first to. So migratory species would be species that live here in winter and here in summer. They go up and down in a mountain, for instance. Nomadic species, they don't have a well-defined home range. And then you have species that are called range resident, that they live in a well-defined region of the space most of their life. And those are the species that are well-defined by this OU equation.
Okay, but you said they have some kind of different kinds of groups. I didn't understand how you said, okay, Palmeiras and
what do you mean? No, so for instance, what George was asking was, in this, for instance, wolves, I don't know, they live in groups of 5, and this group is together, and then each different groups occupy different territories.
And the 5 is family relation, or what does
there are social relationships involving depending on the hierarchies of the species. So I don't know, in some species, this group has only one male.
I understand. And that's the typical structure you need, or is that?
No. All you need is the Austin-Uhlenbeck equation describes the movement of one individual.
I understand, but for example, the one individual next to the group member, that's not described by that, right?
Yeah, so these two individuals are going to be described by OU processes with very similar parameters.
Okay, so you say that also is described. Okay.
The only thing is that if you, okay, so models by definition are wrong, no? So basically, if you want to understand things like what happens when these two guys get very close to each other, for instance, because they are very aggressive, then of course the OU process, if you assume that the movement is not correlated, is not good, because you need some sort of repulsive force But you can incorporate that as some other function, and you can refine all the thing. Usually the OU model is a good descriptor of the movement behavior of these species at long time scales. So when you have data collected for 3-4 years, if you want to fit data for 1 day, then it is not so good, because what the OU does well is to reproduce, when you calculate the variance of C as a function of time, in the OU process, it looks like this. And when you calculate it for your trajectories, it looks like this in the long term as well. In the short time scale, the OU process is a very bad descriptor, because it's linear here, and in data it is not linear. It is, well, it's non-linear.
Hi, Ricardo. Thank you for the talk. Do you have tracking measurements for the first passage problem that you mentioned that you can compare to?
No, we have trajectories of snakes in Thailand that eventually were killed by cars. And we have also data on the death, for instance, for animal-cars interactions, usually the kind of data that we have is more demographic data, so about the populations. So we have things like the killing rate of a given road through different years. And then we also have instances in which there were interventions in the road and how the death rate changed. So the closer connection we want to do with data in this project is to parameterize this PK using, because this PK here at the end can be the traffic flags, can be if the road has lights or not, if it has fences, if there is, it's anything that determines the death rate. So there is data in which they try to measure all these effects, one separately. So one thing we are trying to do is to parameterize this PK, and then the questions that I think, or at least at this point I find more interesting, are related to understanding, as I was saying to Nathan, if these animals are nomadic or if they are range resident, which kind of species are more likely to be killed by human interventions. And this is something we started working in Mongolia in a project that I started doing my PhD. We are trying to understand the movement behavior of the Mongolian gazelle. And the Mongolian government wanted to build a railroad crossing the whole steppe. And they wanted to know how that would affect the area requirements of these animals. So, for instance, I don't know, if the animals have a well-defined territory and the road goes through the territory, then the killing rates are going to be much higher than if it's in the extreme of nomadic species range. So I don't know, what I find interesting about this program is that there is no theoretical framework for it so far. So Benjamin introduced the first one. So that's why, I don't know, I like it a lot. Also, I like first passage problems.
Other questions? Actually, I have a question. I would like to ask your opinion about the following. I have always wondered whether there would not exist some particularly interesting problem that would be could be solved in Brazil because, you know, of our ecosystem is large, the forests, etc. Whether there would not exist some particularly interesting problem to be solved in Brazil because of our forests and so on and so forth. So in such a way that we could take advantage of the local ecosystem to solve some particularly important problem in ecology.
I think every possible interesting problem for an ecologist is in Brazil. Really? So I would turn the answer to say I don't think there is no interesting problem that you can try to solve regarding ecology that you don't find an instance in Brazil. I mean, for instance, we work with people from the Instituto Chico Mendes de Conservação. They have data on jaguars across different biomes in Brazil. The vegetation patterns that I show in the beginning, we haven't found them because I feel bad if I spend a lot of time every day in Google Maps scrolling down the screen, although that is work. But the whole arid region in the northeast has the potential to have a lot of them. The problem is that every year there is people documenting these patterns throughout the world, and so far, I don't know, I guess that because in Brazil, the Amazon is much more interesting than the Northeast for, I don't know, for many people. I meant in terms of ecosystems, not, I know the Northeast is very good.
Well, in the sense that Chile takes advantage of their mountains, but I think we don't take advantage of what we have to lead some areas in science that could be.
I think a big problem for a long time is that there is many people that come from abroad to do that, and that is horrible that I say that because I'm from abroad, but at least I work here. No, but I don't know, for instance, when I was in the US, many of my friends there, they came to Brazil to do field work, and the ecosystem was studied, but it was studied by ecologists from Princeton.
So this is a.
But that happens in many.
We are failing, I think, on this. I think we should
I think the
have stronger groups here working on this.
I think there are very strong groups. I've met very interesting people here, online, all of them, but there is a lot of interesting work done here, I would say. Of course, the Amazon is probably the most famous outside, but I don't know, Mata Atlantica and Desertao, and marine ecosystems as well, sorry. I don't know, and I think there are very good ecologists here. The ones I met, I like them a lot, I don't know.
Okay, so.
But I'm a physicist, so I cannot compare with ecologists from other places. I only know ecologists from here and from the US.
I see.
So,
any other questions? Yeah.
Yeah, what are the assumptions on the noise that you have?
Yes. The noise is white uncorrelated noise. So it's Brownian motion.
And if I introduce correlation between the steps, is that useful for something, or?
So you mean using, for instance, so putting color here in this stochastic force?
Well, you said that this for different times the chi's are not correlated,
yes.
right?
So the correlation function of this noise, it's a delta.
Yeah.
So your question is, what happens if I don't know, I do something like this and I put a characteristic time scale?
Yes.
I mean, it is going to affect the results, no? Because, I mean, with this you change the trajectory. All I'm doing basically is to take tau going to 0, no? The characteristic time scale of the correlation going to 0. But yeah, that correlation time scale might be important. It is not something it is not one of the directions we are extending, but it might be important. Yes.
And are there other potentials that you analyze instead of the OU force?
Yes. So, for instance, there is. So basically, there is two things. One thing is if you assume, and this is something actually I was trying to do in the pandemic. If you consider that this force is not linear, for instance, this force basically is telling me that when the animal is in its center, in lambda, he doesn't feel any attraction to it, but as it moves far away from it, it has some linear force that is going to restitute to recover the position. This is something that I started trying to do was to consider two trajectories, C1 and C2, and here to consider some force that is a function of the distance C1 minus C2, to try to model what I was saying to Nathan, the fact that, I don't know, a prey, when he's very close to the predator, there is some force that goes through it. The problem is that if this force wherever you put in the deterministic term of this stochastic differential equation, if it is nonlinear, then you cannot do anything, but you lose a lot of analytical tractability because nonlinear stochastic differential equations are very hard to treat. Basically because when I put, if this force is linear, this transformation is conserving the fact that if G is normal, then C is normal as well. It follows a Gaussian distribution. But when this force is nonlinear, that is not true. And then the moments of the, if I am using too technical words, just tell me I don't know what a moment is, and then I change the words. But basically the idea is that the mean, the expression that I have for the mean and for the covariance, they are coupled to each other. So I cannot treat the stochastic process analytically as easy as I can do when this is linear and the process is a normal Gaussian process. It's a Gaussian normal, a Gaussian stochastic process. So this is one thing that we are trying to do, to incorporate forces that do not attract the individual towards some fixed location, but they interact to each other. They attract to each other, but that is hard. And another model that there is another model that is better to describe animal movement than this one, which is, so basically, if you translate this equation to physics, this is an harmonic oscillator, a particle that is trapped in a harmonic potential with some thermal fluctuations. No? But this is in the, and this, I always say this wrong, this is in the overdamped limit, right? I am neglecting any, I am basically neglecting any characteristic time scale in the relaxation of the velocity of the particle, right? So, a better model to describe animal movement has an equation for x, and then it has an equation for v with some relaxation time scale of the velocity to some equilibrium.
Without noise, the second one.
No.
Oh, with noise? Okay.
With noise, yes. And then, here you have v, and then you have the linear force, I think, as well, yes. So basically, this is the same as that equation, but I have an additional time scale. And basically, when I do this, when I use this model is very good also at short time scales, basically because the idea is that it is also describing the autocorrelation of the velocity. So the general picture is, if you want to understand the behavior, the properties of the trajectory at long time scales, that is fine. OU is fine. If you are interested in short time scales, so something, for instance, that I am trying to start thinking of is whether, given a trajectory of an animal, I can understand what is the animal doing just classifying basically behaviors of the animal based on the way they move. So if the animal is, of course, if it's sleeping, it's very easy. But if the animal is looking for food, or it's looking for a mate, or in the case of, I don't know, sexual species that reproduce sexually, whether it's in, I was going to say something barbaric. If it's in the breeding season, or if it is not. So trying to infer the state of the animal, looking at the way it moves. And to do that, very often you need high resolution data in short time scales, and then you need that model. So basically, there are, at the individual level, accounting for autocorrelation is the natural extension. When it goes to populations, accounting for cross-correlation terms, it's the natural extension as well.
So any other general question? So I think it's time to finish. Let us thank Ricardo again for this talk.
Thank you.
I think those who want to make other questions, please go ahead and
Sure.
ask him privately.